the advancement in our understanding about plant growth and recent technological developments, a new chapter called biotechnology has evolved. One of the most popular and applied in the biotechnological field is plant tissue culture. It has been recently included in our postgraduate and undergraduate courses of biological sciences.
Since the beginning of agricultural practices, man has been trying hard to improve crop plants and increase their yield. This was done by traditional method of plant breeding. However, present progresses made in the field of plant physiology and genetics made it possible to evolve a scientific method for increasing crop yield and crop type. For the selection of desired traits, a modern scientific method has been evolved. This depends on a new procedure called tissue culture. In this method, plants are grown under controlled environments to suit our desire for their growth. At the beginning of this century, a famous botanist called Haberland presented his beliefs that a plant cell or a tissue can be regenerated into whole plants, intact plants. This was tried to be proved correct by many scientists, but they failed because of inadequate knowledge about the axenic cultural practices and nutritional requirements of isolated plant parts. In 1939, two famous scientists, Godrej and P.R. White, published separately very convincing reports of cell growth from carrot cambium cells and from tobacco tissues into shoots. Their work has proved that plant cells have enough genetic information and physiological machinery to develop into an intact plant. And this capability of a cell or tissue is called totipotency. The whole tissue culture is based on this belief and this capability of plant cells.
There are two predetermined plant structures. One is mature or immature zygotic embryo, and the other is shoot-tip meristem. If these two structures are placed in vitro conditions, they can develop into whole plants under the influence of different plant growth hormones. But if any differentiated tissue like leaf or petiole is used as explant, they begin to proliferate into an undifferentiated mass of cells called callus. These callus cells, under proper hormonal environment, creates differentiation of shoot bud or somatic embryo, which eventually develops into intact plants.
A standard tissue culture laboratory should provide following facilities. One is a storage space for chemicals, glassware, and prepared stock solutions, nutrient media, etc. Washing facilities for very intricate washings. Also storage of stock solutions, etc. under frozen or deep frozen conditions. There should be sterile conditions for preparation of media, and there should be place where septic manipulation should be done, and culture conditions where the cultures are stored. That room should have enough environmental manipulations for their growth, and above all, a condition for evolution of growth in aseptic conditions should also be there.
The first step in proper washing of glassware is that glasswares are washed in detergent solutions. Then they are soaked in concentrated sulfuric acid and potassium dichromate solution. Automatic dishwashers in big labs and ultrasonic washers for pipette and test tubes are recommended. Tap water is unsuitable for washing. Therefore, glassware should be washed first in tap water. After that, they should be washed in demineralized water. And then the third step, they should be washed with double distilled water. Nutrient media for tissue culture should be always prepared with analytical grade reagents or chemicals. Weighing of chemicals is done on analytical balance. The first step in media dilution is agar melting. Agar at a concentration of 0.6 to 1.2% using double distilled water is slowly melted using water bath. Sucrose is a universal carbon source for plant tissue culture. It is added at a concentration of 2 to 5% to the media. There are two types of mineral requirements: macro, which is required in a high concentration, and the other is micro, which is required in a very small amount. Macro elements are nitrogen, phosphorus, sulfur, calcium, potassium and magnesium. For proper development of cells, 25 millimole of nitrate and potassium is required. There are 6 important microelements: manganese, zinc, copper, boron, molybdenum and chloride. Concentrations of both elements varies according to the media composition.
Iron is used in chelate bound form. NaEDTA is the standard chelator used in plant tissue culture media. Ferric EDTA remains available to the plant regardless of any pH.
Vitamins are an important growth factor. For example, thiamine, a vitamin B, some vitamin B complexes like nicotinic acid, pyridoxine, pantothenate, etc. is also added. A higher concentration of inositol, that is 100 ppm, is added to stimulate plant growth.
Now we come to the most important cell differentiation factor called plant hormones. There are 3 types of plant growth hormones which are used in tissue culture. One is auxin, like IAA, IBA, NAA, and 2,4-D. Auxins are used mostly for initiation of cell division and roots. Another type of hormone is cytokinins, which are adenine derivatives, and they are used for shoot induction and propagation. They are kinetin, BAP, 2IP, and zeatin. Another hormone, gibberellic acid, is used for promotion of cell growth.
Moreover, some common ready-made media are available commercially. For example, Murashige and Scuve media, Gamberg's B5 media, White's routing media, etc. All the above-mentioned ingredients are mixed in desirable amounts and made up to one dilution mark with the help of double-distilled autoclaved water. pH of the media is adjusted by adding low concentrations of NaOH or HCl. pH meter is used for taking readings. pH determines the solubility and availability of mineral ions. pH of a plant tissue culture media ranges between 4.5 to 6. Media prepared with all the mentioned ingredients and hot agar is poured in culture vessels and sealed with cotton bunks and aluminium foils. For meristem and embryo culture, test tubes have been found more convenient. Whereas for callus growth or cell suspension culture, flasks have been found more suitable. Sterilization of media is done by autoclaving. Autoclaving time varies with the volume of the media to be sterilized. For example, 20 ml volume needs 15 minutes of autoclaving at 1.5 kg per square inch. The autoclave pressure is brought down after the required time. Now, as the pressure becomes normal, the media is removed. Media is removed from the autoclave and kept at an angle to prepare a slant. Alternatively, filter sterilization of thermolabile compounds is done. For this, 0.2 micrometer pore size, bacteria-proof filter papers or membrane are used. Filtration membranes are put on the filter holder. The filter holder, along with a flask, is then wrapped in aluminium foil. And the whole assembly is then autoclaved. Autoclaving is done for 15 minutes. After autoclaving, the whole assembly is removed. The filtrate is then gradually drawn through the membrane into a sterile container. All this is done under laminar flow. Then filtered hormones are ready to be added to the prepared media.
In this episode, we have tried to introduce you to a subject called plant tissue culture, which is based on the principle of totipotency. We have also presented requirements of a plant tissue culture laboratory, what the laboratory should be like, how the aseptic conditions should be maintained, what are the nutritional requirements of an ex-plant growing as a tissue, and also the requirements of their growth, and we have tried to tell you how a standard tissue culture media is prepared. We could not introduce the whole subject in detail to you, but in our next episode we would try to tell you how to introduce an ex-plant to the nutrient media for their growth.